Just a couple of things before we start. First, I don't want to know that has been alcohol distributed in Biomolecular. Before the lecture. Even after the lecture, I will do. Anyway. Second, this is the first time our Christmas lecture will be tape recorded. Then, well, this is our plan to some of the activities, the scientific activities we do in Biomolecular, we want to start tape recording so that we can have an archive of these activities, so that we are able to have a memory of our activities, and eventually we will be in YouTube, so you will become famous and if rich. So this is our plan, to tape record a number of the most important activities we do every year. And I think this is a very important activity, this Christmas lecture. Manuel and I really think this is a very important thing we do every year, and we come together. And I'm very pleased and happy that you all came, even if it's not going to be a wide party this time, because of the economic situation. So the program today is, as usual, with the Christmas lecture. But I think that in addition to tape recording, this time there's going to be a Christmas lecture with an experiment. So this is very brave from you. And then we will have, as every other year, we will give, you're passing drinks. What I was saying. Oh yeah, then we will have our diploma to all the people that finished the PhD degree this year on previous occasions. So for the introduction of that will be Manuel Thomas. I don't want to know what type of liquid you are handling there.
Good morning. Even though we were here in Pune, and according to previous years, Jose Maria had to make the presentation of the introduction of Ralf. We have decided on the spot that I will do that this time. And for me, it's a real pleasure to introduce Ralf Richter, who is one of the brilliant young researchers in the Biomolecular. Ralf is the kind of person you don't know whether he's a chemist, a physicist, or a biologist. So I understand, if I remember well, that he got a degree in physics in Germany. Then he has some experience in industry in Sweden, some academic experience also in Sweden, in Göteborg, with Professor Casemo, who is one of the leaders in surface science. And then he came a little bit down, and he went to Göteborg for his PhD thesis with Professor Anderson. It is also a leading microscopist. So then he went to his country, to Germany, and he was working in Heidelberg at the Max Planck Institute in Heidelberg, and then in Stuttgart. So, and finally he decided to come with us, because I think it was a very nice thing. And he's one of the biophysicists in the biosurfaces units in the Biomolecular. So he will give today a very nice lecture, I'm completely sure. You only have to read the title. How to then get into the board. Okay, Ralf.
Thank you very much, Manuel, for the introduction. It's of course a big honor for me to speak here today. It's also a surprise. I did not expect to be solicited to give this lecture here. I was thinking, after all the encouragements last year, that there would be a woman standing here. That's certainly not me. But I still will do my best, and really I'm pleased to stand here. I'm a quite conventional person, I think, so I was hoping I could avoid in my lifetime to appear on YouTube. I will see how this goes. But anyway, what I will try to entertain you with today is jelly. What that has to do with our research, I hope you will see and feel throughout the talk. Just a little historical note. This sentence here, nailing jelly to a wall, has been phrased by Theodore Roosevelt, the second president in the 20th century of the United States of America. That was by the time when they wanted to have the Panama Canal built, which by that time still went through area of Colombia. And in a letter to his friend and former classmate and writer, he said, "You can no more make an agreement with those leaders of Colombia than you could nail current jelly to a wall. And the failure to nail current jelly to a wall is not due to the nail, it's due to the current jelly." So the sentence means that there's something that's very, very difficult to achieve, nailing jelly to the wall. It turns out that Roosevelt, well, was not only a politician, he was also a scientific visionary. I think by that time they had no clue what jelly actually is on a molecular basis. And it turns out that even today it's not easy to understand how these systems work. So these are really difficult to work with. There are some people in the world that try to do it on the microscopic scale. As you see here, even with many nails, it doesn't seem to be so easy. So that's not the way that we go. Just from a more scientific point of view, what is jelly? The one that I showed you before and the one you have been eating contains around 10% gelatin, probably a little bit of sugar, I don't know how much, but also most of it is actually water. And as you may have experienced when eating it, it's certainly not a liquid, at least when you have it in the beaker. But you may also have realized when you put it into your mouth and you keep it there for a few seconds, that it starts to change its properties. It has been a soft but solid material before, and it starts to melt and become a liquid just because you put it into your mouth. So that's, I think, a very neat illustration of how subtle you can tune material properties in these type of jelly or hydrogel-like materials. So what my talk will be about is physical hydrogels. Jelly is one of those. The definition would be of a gel would be a polymer meshwork. A physical gel is a gel where you have either crosslinks or just entanglements between these polymers, and that these crosslinks are reversible. So they can open and close quite easily. Here you at least have clear crosslinks. Here it's just entanglement. There the polymers are wound up into each other, and these entanglement points can actually act pretty much as if they were transient crosslinks. So it's not such a difference between this and this situation. If you think about it, well, and hydrogels means that all this is in water, and typically, as shown here, you really can entrap tremendous amounts of water inside these structures. The density of the crosslinks will define a certain size, a mesh size. One mesh is schematically shown here. And you can imagine all this is flexible, all this is opening and closing, so there are a lot of dynamics, in particular dynamics. And the dynamics and the properties of the polymer that you use here and the properties of the crosslinks give these materials very special mechanical and partly also chemical properties. So, jelly in biology, I would like to term the materials that I will talk about biomolecular hydrogels. One example is shown here. This is Ram yesterday and the day before. He was quite sick, and you may realize that he is throwing away a napkin containing some slimy fluid or slimy liquid, and he's quite careless in doing so. When you do the type of research we're doing, you start to think about this in a different way. Mucus is one of the prime examples of biomolecular hydrogels, and you can produce about 1 kilo of it per day. And it's important. I mean, it's not for the reason that when you're sick you produce a lot of it. On a smaller scale, on the cellular scale, you can find such hydrogels outside the cell as coats, so-called pericellular coats. You can also find them inside the cell. This is a very small microscopic gel which is in the inside of the pore that links the cytosol and the nucleoplasm to each other. I will not talk about RAM today, but I will talk about pericellular coats, and I will talk about the nuclear pore complex. And the main goal with all this is to understand how do these biomolecular hydrogels work? What's the mechanism? So I will start with the extracellular world and how to enrich pericellular coats. And I will give you one example. And this example is important for survival of any mammalian, almost any mammalian species, I should say. What you see here in the middle is an oocyte, it's an egg cell. And around that egg cell, you saw this matrix growing. And this matrix is called the cumulocell oocyte matrix, or the whole thing is called the cumulocell oocyte complex. You have not only the egg cell here, you have many small cells around it, which initially were tightly attached to this egg cell. But they produce something. They produce actually sugar, hyaluronic acid, and they produce a few proteins. And these proteins assemble into a very soft, large matrix that can reach the size of several times the egg cell's diameter. Well, there are some proposals here of how this type of system may be stabilized. You see, these are names of proteins. You don't need to remember them, at least not right now. What I would like to point out is that if you remove one of these proteins, this matrix is no longer stable. It will just disintegrate, and if that happens, you're no longer fertile. So stability of this coat is critical for fertility. To give a little bit more of an idea of where this coat comes into play, here's a journey of the egg towards a key step, fertilization. It starts off in the ovary in a follicle. You have the oocyte with this compact cumulocell matrix. It's expanding. That's the process that I just showed you in the movie. After some trigger, it's expanding for about 12 hours. And then the follicle ruptures, and that's what is called typically ovulation. The oocyte is then ejected from the follicle. It's sort of attached, it's picked up by the oviduct. It somehow travels and is transported by the oviduct into the oviduct. There's a narrow orifice in the beginning, and then at some point, once it is in the oviduct, that's the place where, if the egg cell is lucky or if it wants to be lucky, can be fertilized. And each of these steps, here, 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 and here, it turns out that this coat is important. The oviduct is quite a special thing. It's a sort of channel which is coated with cilia. Cilia are membrane, finger-like protrusions from the cell membrane, and they move in a periodic manner. And you can imagine if all these, they're moving pretty much in the same direction, and this makes actually these oocytes moving into the oviduct. And I have to give you an idea of how important mechanics is. I have two movies here. Let's see if they work. They don't. So this is the oviduct, that is the entrance, and this is the cumulocell oocyte complex. What is stained here is the nucleus of all the cells. So you don't actually see the matrix, but you see everything is connected, and that's the matrix. And you see how soft it is and how now it actually starts to be moved in here, and it pops off here, and then little by little is really wiggling into the entrance. So it's a really soft thing, and that softness seems to help it to move along that surface. If I would do the same with an oocyte that has been perturbed a little bit, you see that the matrix is much stiffer. It adheres and moves along here, but then it just goes off, and it will never enter into the oviduct. So the material properties of these codes clearly are of primary importance to understand for these things to work. So let's go to the molecular level. The protagonist is a sugar, it's hyaluronic acid. It's quite a simple sugar. It's a polymer of disaccharides, glucuronic acid, which is charged, and acetylglucosamine, which is uncharged. This is about 1 nanometer. The molecule isn't known to have any except one chemical modifications in nature. It's always linear, it's unbranched, it's really simple for a sugar. The only thing that varies a lot is the degree of polymerization. You can have up to 25,000 of these units, which means that the molecule is several micrometer in length. It's a polymer. It's a large polymer. If you look at this from the polymer physics point of view, you could say that hyaluron is well described as a flexible polymer, or because it's charged, a flexible polyelectrolyte. And that's a really important, apparently simple but really important statement because it has implications. A flexible polymer, imagine spaghetti. Uncooked spaghetti will be quite stiff. So this is not a flexible polymer. Cooked spaghetti will be quite flexible. You can actually, if you look at a single chain, you can determine that there is a certain characteristic curvature in the bending of these chains. And this curvature is related to what is called the persistence length. The persistence length, so it's a measure for the flexibility. For uncooked spaghetti, it would be 4 meters or so, I would guess. For cooked one, it's a few centimeters. And for hyaluronic acid, the persistence length would be 4 nanometers. This is still 10 times more than, for example, peptide chains would have, which means that if you put this in solution as a polymer, it will just wriggle around and form a random coil, a sort of spherical shape, moving around all the time. If you inscribe it into a sphere, you would find that here there's just 0.05% of sugar and 99.95% of water. If you increase the concentration in solution somewhat, these individual chains, they will interpenetrate, they will form a meshwork, and then you have an entangled polymer meshwork, or one sort of a physical hydrogel. Interesting things happen also, and all these predictions come from relatively simple polymer theory. Interesting thing happens when you put these things on the surface. If you attach them with just one end at the surface at low concentrations, the shape will again be very similar to the one in solution. You have a sort of random coil. This is called the mushroom regime. If you go to higher concentrations, these individual chains, they will repel each other, and the only way they can avoid contact is actually by stretching into the Z direction, and this is called a polymer or polyelectrolyte brush. And these things just happen because it's a flexible polymer and it's confined to a surface, like, for example, the cell surface. That's one of the key properties of hyaluronic acid. Another one is that there are a number of proteins that bind specifically to hyaluronic acid. And this, since hyaluronic acid is a polymer, there's not only one protein binding, there can be tens, hundreds, or even thousands of proteins binding. And that gives rise to very different supramolecular architectures. Some of them are shown here. Some of these proteins help actually to attach hyaluron to the cell surface, and others have been proposed to cross-link or to change the properties of hyaluronic acid. And we would like to understand these assembly behaviors and how they give rise to different properties, physical chemical properties and functions. There's a problem here, and the problem is that these things are extremely hydrated, they are disordered, they are large. That's typical properties of biomolecular hydrogels, and they're dynamic. And if you think about the conventional methods like X-ray, NMR, electron microscopy, or other biochemical methods, it's not so easy to look at these systems directly on the supramolecular level. What people have done in the field, they have looked at the molecular level. We have now a few structures which show us clearly how proteins interact with HA on the atomic scale. People have looked on the cellular level and tried to understand what's going on. This is often remaining on a rather descriptive phenomenological stage, but there's very little information directly available on the supramolecular level. And this is what we are doing. The approach is to take the molecules that we know are involved, to put them together in a controlled way, and to create supramolecular model systems. And I would like to show you today a little bit how we create those, how we characterize them, and what we do with them. So the starting point is always a surface, silica, for example, and a lipid membrane. This lipid membrane can be prepared from liposomes, from small spherical membrane assemblies by an self-organization process. The nice thing about membranes is that we can mix different lipids together. And if we, for example, want to have an anchor for this group here in red, we can mix it in at a certain ratio. And we can mix in different anchors as well, so we can sort of address or create certain attachment points for other proteins or for other molecules on that surface. At the same time, there's a rather low level of unspecific binding, so we don't have to care so much about passivation of these surfaces. Here is an example of a measurement that we are doing by quartz crystal microbalance. The decrease in frequency here, the blue curve, tells you that something is binding. The increase in dissipation that you see here tells you that what is binding is quite soft. From these curves here, we can tell immediately that these vesicles attaching here, they did not rupture into this type of conformation, they stayed intact. If we see that type of curve, then we know that the vesicles first adhere intact, but at some point they rupture and they form this bilayer that we want to have. So this is our benchmark. If we see that, then we know that our surfaces will behave the way we want them to behave. And here's how we create a model system. I call that hyaluron Lego. That's the formation of the bilayer, as you just saw. These vesicles from which the bilayer was formed contain some lipids with biotin, and the streptavidin, the protein linker, binds specifically here, and that's the second step here that forms a monolayer. And then in the third step, we have used hyaluronic acid with just one biotin group at the reducing end, and from that we know that it should just attach via the biotin to the streptavidin. So from looking at these type of curves, we learn directly something about the assembly mechanism, and we can follow how long it takes to form these type of models. To characterize the systems in more detail, we use a whole toolbox of techniques. I will not go into detail of how we do that, but the essence is that each of these techniques just gives us one or two parameters. If we combine everything, we can get a detailed picture of what these films are looking like and the properties they have, and we can also avoid artifactual interpretations of the data. From a combination of QCM, ellipsometry, and microinterferometry, for example, we can conclude that our films, these hyaluron films that we form, have a thickness between 120 and 500 nanometer. That depends a little on the molecular weight of the hyaluronic acid that we use. And from these numbers, we know also that these films can be described as polymer brushes on the surface. As I mentioned before, we know also quite well what the average spacing or what the surface density of hyaluronic acid on the surface is. And if we make all the calculations, we find that this example here, for example, we have 99.8% water in this film and just 0.2% of hyaluronic acid. So what can we learn? One example is the interaction of TSG6 with such films. TSG6 is one of the proteins that is important for the stabilization of the COC matrix. It contains two parts, a link module and a cap module, and the hyaluronic acid is binding to the link module. So we have created our hyaluronic acid films and then injected TSG6 as a start. To keep things simple, we just used that part of the protein, not the entire protein. What we found was it binds, and if you rinse, it unbinds. It's a fairly weak, but it's a detectable interaction. And that was pretty much consistent with literature data on oligomeric sugars. We had a big surprise when we then added the full-length protein. Here we get a much stronger binding, and also the desorption is much lower. So apparently the full-length molecule binds much stronger than just the link module, indicating that the second part, the so-called cap module, must somehow be involved in the binding. We did then some kinetic analysis. That's a typical titration curve by ellipsometry, where we measure the amount of adsorbed TSG6 as a function of TSG6 in the bulk solution for both linked TSG6 and full-length TSG6. And these are so-called titration curves. This is a typical curve that can be fitted well by what is called the Langmuir isotherm. From that we get a KD, 5 micromolar. This curve here is more intricate. It's this S shape or sigmoid, slightly sigmoid, tells us that something special is going on here. That's a fingerprint of cooperative binding. And if we do what is called a Hill analysis, we get a Hill coefficient of 1.8, and we also get a KD that is about 10 times lower than the KD here. So we have two contributors to an improved binding, cooperativity and in decreased affinity constant. Why this cooperativity, we were asking, and what is the effect that TSG6 actually has to the films? And to check that, we measured the film thickness as a function of the amount of TSG6 in the film. Without TSG6, we get 400 or 500 nanometers, as I mentioned to you before. But then if we add more and more TSG6, the thickness decreases. So we actually get a collapse induced by the addition of the protein. And I'd like to point out that the physiological concentrations of the protein are somewhere here. So even under physiological conditions, you would expect to have these drastic conformational changes induced by that protein. We were wondering what's the mechanism, and one that of course comes to mind is that TSG6 acts as a crosslinker, and these crosslinks sort of lead to the collapse of the entire film. We mentioned that we managed to demonstrate that directly in a slightly different assay. Here you have now very short chains of hyaluronic acid. They're so short that you can only bind a single protein to each of these chains. And that's the binding curve here. And then if we add on top of that hyaluronic acid as a polymer, we see additional binding. And this additional binding tells us that even though TSG6 is bound here, it has yet another binding site available for binding other hyaluronic acids. So that's a direct indication for cross-linking by TSG6. So that's quite neat. We were thinking that may be one explanation of why TSG6 is necessarily in stabilizing the COC matrix. It forms a cross-linked meshwork, but it also collapses the film, and that's something that may not be so good for the COC matrix. One has to keep in mind that TSG6 is a very promiscuous and polyvalent molecule. It binds to different things, and apart from binding to HA, it does also act as a catalyst in another biochemical reaction. Inter-alpha inhibitor is a very special protein that proteoglycan, which contains convertin sulfate chain and then some peptide chains attached to that. And TSG6 can actually catalyze the removal of these heavy chains, HC. It forms a complex, and then this complex can be opened up again by interaction with HA, and at the end you have a covalent complex of hyaluronic acid and heavy chains. So here TSG6 is a catalyst that triggers a covalent trans-esterification from the inter-alpha inhibitor to the hyaluronic acid of heavy chains. And we were wondering if it has these two roles. Well, what does it do if you present these different molecules? Does it do both at the same time or just one thing? And here's a comparison. That's a mixture of TSG6 and the inter-alpha inhibitor, so the substrate for the enzymatic reaction. And this is TSG6 only. The same amounts of TSG6 were used here, and you see directly that binding here is very fast, whereas here you have something going on, but it's a much slower process. Indicating that two different things are happening here. Probably this is a heavy chain transfer, and that this is really heavy chain transfer is shown if we add a cheiotrophic agent, guanidinium hydrochloride, which would remove all the non-covalently bound material from the film. We see that there's very little desorption here, so these are most likely heavy chains, and whereas there's substantial desorption here, that is most likely TSG6 bound into the film. So from the fact that we don't see fast binding here, we can conclude that the complex formation of TSG6 and heavy chains is actually impairing the hyaluron binding activity of TSG6. So we have here really an interplay of different processes. TSG6 function depends strongly on its environment. If there is inter-alpha inhibitor present, it will do this type of work. If there's only hyaluronic acid present, then it will crosslink and collapse the film. And this is pretty much where we are with that story right now. There are a number of additional players here, which I will not talk about, but I hope that within the next years we get some decent idea of how the COC matrix is stabilized. This brings me to the second topic of the talk, and that's the inside of the cells, the nuclear pore permeability barrier. Here's a little bit of a background. You have the whole slide is the cell. You have the nucleus somewhere inside the cell. It's embedded in a double membrane, and you have the cytoplasm around. The DNA is here. The ribosomes are here. So one has to find ways to get the RNA out, but one also has to find ways to get proteins that are produced here again back into the nucleus. And all this is done via one single type of channel, the so-called nuclear pore complex. It's an opening that is around 40, 50 nanometers in diameter, which is large compared to protein sizes. If you would have such a channel just open, everything would go in and out, and you would have equilibrium within a very short time. To prevent that, these pore complexes are filled with some peptide or protein stuffing, and that stuffing resembles actually a gel to quite some extent. This is an artistic presentation by another researcher of how this could look like. The idea is that you have some sort of entangled meshwork of intrinsically disordered proteins sitting inside this pore. What it does is it prevents the bad guys from going in, and somehow the good guys have to find ways to get through. And the way they do that is that they attach to what is called a nuclear transport receptor, and this receptor carries it through in a way that is not entirely understood, and which we would like to understand better. So what is known is that these intrinsically unfolded proteins that are sitting here have a very specific amino acid sequence. They're called so-called FG repeat domains because they contain repeat domains that are rich in phenylalanine and glycan. And these FG repeats are the recognition sites for these transport receptors. They help them somehow to get through. And we know also that these are attached via one end to the channel walls. So again, You can think about it as a brush or as a gel. There are a few models around of how this works. One of them is the so-called selective phase model, and in this model it's proposed that these Fg repeat domains, they form a gel. Actually the Fg units, they are slightly hydrophobic, and maybe there are other interactions as well which make that they cross-link transiently so that you get a sort of gel here. Nuclear transport receptors, they have binding sites which compete with these cross-links, so they sort of can open up the individual links and then melt through the gel. Other probes that cannot interact, they just see this meshwork here with a rather small mesh size, and they cannot enter and not penetrate efficiently through it. A support for that model was that if they purified large amounts of this compound, they could actually produce macroscopic gels, as you see here. What you see here, we were actually wondering, could we sort of reproduce such a type of selective behavior in a macroscopic system? And my students and my people have been quite busy in the lab doing macroscopic experiments, and Nico will have one trial to demonstrate you what we have been doing. So these are now two samples of jelly, pretty similar to the one that you have been having earlier today.